Hello everyone. Welcome to today's webinar. My name is Dr. Shweta Pahujani and I am a senior product manager at BioVision. Today I will be talking about BioVision's ELISA kits portfolio. Please feel free to type if you have any questions in between. Otherwise, we will be taking Q&A section. We will be having Q&A section at the end of the webinar. So let's get started. In my presentation, I will be covering following topics. So I will talk about a general introduction or overview of ELISA principle. I will talk about different types of ELISA formats available in the market. Then I will talk about how the characteristics of an ideal ELISA kit. I will share some tips for choosing an ideal ELISA kit for the end users. Then I will talk about our BioVision ELISA kit portfolio, or the different kinds of or different categories of ELISA kits that we offer for various research areas. I will share some experimental tips for our end users, and at the end we will be having our Q&A section. So let's get started. Okay, so here is a brief introduction of ELISA. So, enzyme-linked immunoabsorbent assay, which is commonly called as ELISA, is a common immunoassay in which antibodies, peptides, proteins, and small molecules can be detected and quantified using a multi-well plate. Usually it's a 96-well plate. The common principle on which ELISA assay is based is it includes immobilization of an antigen or antibody to a solid phase, followed by a separation of bound and free reagents by washing steps. The readout or the signal can be, the readout, which is a signal. It can be detected either, it can be in the form of either colorimetric, fluorometric, or luminescent signal. The ELISA procedure results in a colored end product, which correlates to the amount of analyte present in the original sample. So this is the common principle on which all the ELISA assay is based on. So there are different kinds of ELISA formats available in the market. So commonly there are 4 different kinds: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. I'm going to talk about the principle of each types. I will explain how the principle of each different kinds of these formats. I will be talking about the application, how one can select which kind of format is applicable for which application, and I will also talk about the advantages and disadvantages of these ELISA formats. So let's start with direct ELISA. So direct ELISA is based on following principle. The antigens are immobilized on the surface, and the enzyme conjugated primary antibody are used to detect or quantify the antigen concentration. So this is a very simple or straightforward method. We see the antigen is immobilized on the surface, which is in the 96-well plate, surface of 96-well plate, and an enzyme conjugated primary antibody is used to detect or quantify antigen concentration. In this kind of ELISA, the specificity of the primary antibody is very important. Some of the advantages of this kind of ELISA is it is a very quick procedure. Very few steps are involved, and the cross reactivity of secondary antibody is avoided, as only one antibody is involved. So there are low chances of cross reactivity. Some of the disadvantages are It requires labeling of all primary antibodies. Immunoreactivity of the primary antibody may be reduced as a result of labeling. Labeling primary antibody can be time consuming and expensive. It provides minimal signal amplification, and it is an expensive assay. Some of the application where this type of ELISA is commonly used is for the titration of conjugated secondary antibodies, as well as it is also very useful to estimate the antigen cross-reactivity. So second type of ELISA format is indirect ELISA. And the indirect ELISA is based on the following principle. In this, the antigen is immobilized on the surface. Primary antibody, which is not labeled, but it is detected using a enzyme conjugated secondary antibody that recognizes the primary antibody. So two types of different antibodies are involved, a primary antibody and a secondary antibody that can detect, or the primary antibody, secondary antibody that can detect a primary antibody. Some of the advantages of indirect ELISA are, so it is more flexible and versatile assay. Unlabeled primary antibodies retain maximum immunoreactivity. It provides higher sensitivity and higher signal amplification. Some of the disadvantages are cross-reactivity may occur as with the secondary antibody, which can result in non-specific signal and provides a high background. It requires an extra incubation step. So the overall assay is relatively longer as compared to the direct ELISA. Third type of ELISA format is sandwich ELISA. In this type of ELISA, the antigen of interest, that is which we want to measure, is bound between the capture and detection antibody. The two antibodies used in this assay must be very critically chosen to prevent cross-reactivity or competition of binding sites. So two different kinds of antibodies are involved in this assay, and one is capture antibody and another is detection antibody, and the antigen is bound between these two antibodies, and then the secondary antibody is added, that is conjugated antibody, to generate the signal. So the more important one important application for which this kind of ELISA can be applicable is this system allows multiple sample evaluation using different primary antibodies to be screened with a single conjugated secondary antibody. Some of the advantages of this ELISA format are, it is very sensitive, it provides high specificity and sensitivity, it is suitable for complex samples, and it is very flexible. The assay is very flexible due to the use of two different antibodies. Some of the disadvantages of this kind of ELISA format is antigens must contain at least two antibody binding sites. It is a relatively expensive assay as two different antibodies are involved. So it added up the cost and the development time. And it involves a longer, many incubation steps, so longer processing time with multiple incubation and washing steps. So the fourth type of format, ELISA format, that is available in the market is a competitive ELISA. So this is the most commonly used ELISA format. This method is usually used when only one antibody is available, or the target protein or the analyte is very small. The principle of this format is the antigen of interest and purified immobilized antigen compete for binding to the capture antibody. So that means, so the purified immobilized antigen or purified antigen is immobilized onto the surface well and which compete with antigen, a target protein or the antigen of interest present in the sample. And they both compete to binding to the capture antibody. Decrease in signal compared to purified antigen indicates the presence of antigens in the sample. So the higher the the higher the signal, that means lower the concentration of sample or antigen of interest. So it is inversely proportional. The concentration of the antigen in the sample is inversely proportional to the signal. Some of the advantages of this ELISA format are: it measures the concentration of an antigen or antibody in a sample by observing interference in an expected signal output. And most often this kind of competitive ELISA is used when the analyte is small and cannot be bound by two different antibodies as it can be done in the capture or sandwich ELISA. Some of the disadvantages of this ELISA format is it is less intuitive in calculation. It has low overall sensitivity and specificity. As we know, antigens must contain at least 2 antibody binding sites, so it requires higher cost and development time due to multiple incubation and washing steps. Okay, so now the question for the end user is how they can select which kind of format is available for their application. So I'm going to, I'm going to give some tips for how our end user can select an appropriate ELISA format or appropriate ELISA kit for their application. So some of the parameters that we recommend to look while selecting an ELISA kit for a particular application are: so first is sample species. So there are different kits available in the market for different sample species, like human, mouse, rat. So these kits differ in terms of antibody used in the kits. So that means the antibody that is used in the kits is specific for certain species. It can be either specific for human samples, mouse, or rat. So the specificity of the antibody used in the kit makes the ELISA kit specific for certain sample or species sample types. So one should look for so end users, if they know the species of their sample, they can look for the ELISA kit that can be specific for their sample species. This will be also helpful to avoid the cross reactivity. The second parameter that they should look for is the analyte. So the end user should know that what kind of analyte or target protein they want to detect. So for example, sandwich ELISA is generally suitable for detecting large proteins with multiple epitopes, while the competitive ELISA is suitable for detecting small molecules like antibiotics. Sandwich ELISA can detect large proteins like cytokines. So if they know, what kind of protein or analyte they want to detect, they can choose the ELISA kit based on that. So if they have a large protein with multiple epitopes, they can go for sandwich ELISA, and if they have a small molecule that they want to detect in their sample, they can go for a competitive ELISA. Next parameter is the mode of analysis. So ELISA is a tool that can be used for both qualitative and quantitative analysis. What does it mean? So qualitative ELISA provides only simple result, that is a positive and negative result in the sample. So it only gives the information that whether the target protein is present in the sample or it's not. while the quantitative ELISA can quantify the concentration of the analyte or target protein in the sample by comparing it with the standard curve. So if the end user, they know, if they want to only know that whether the analyte or the target protein is present in the sample, uh, and they do not want to quantify it, they can go for qualitative ELISA. But however, if they want to know the exact concentration of their target protein in the sample, they should opt for quantitative ELISA. Okay, so next parameter for choosing an ELISA kit is the sensitivity. Okay, so sensitivity is it is a very important parameter. So, for example, if the user, they do not know the concentration of the target protein or analyte in their sample, they can always go for the ELISA kit with a broad detection range. And if the concentration of the analyte in the sample is very low, then ELISA kit with a high sensitivity are always recommended. On the other hand, if the concentration of the analyte in the sample is very high, then it is always recommended to dilute the sample so that the concentration of the analyte is within the range of the standard curve of the ELISA kit. So the base is the sensitivity. That means they should look for high sensitivity for low concentration, and they can always dilute their samples if the concentration is very high, so that it can be within the range of the standard curve. Next parameter to choose an ideal ELISA kit is a sample size. So in general, the ELISA kit requires as low as 10 microliter sample and as high as 100 microliter. So the range is between 10 microliter to 100 microliter sample for analysis. So if the amount of the sample that the end user want to analyze is very small, or for example, they have a very precious samples, like for example, in vivo samples. So it is always better to choose ELISA kit that requires less amount of samples. So this way they can, they can definitely save their precious samples, because if they have to go for ELISA that requires like 100 microliter or more, so then they can definitely, it's not a good choice because they will end up using all their samples. So next parameter is recovery and linearity. So what does it mean? Recovery and linearity experiments are used to assess the performance of ELISA kits. So what does recovery data tells us? Recovery data will help to determine whether the analyte detection is affected by the differences in sample matrices. So samples can be in different matrices. So the recovery data will tell whether the analysis is affected by the different the mode of analysis, or the the performance of the kit is affected by different matrices. So the higher the recovery, the better the ELISA kit is. While the linearity of dilution determines the extent to which the dose response, meaning the different dilutions of the target protein is linear in a particular diluent. That means if the different dilutions of the protein of interest, or the analyte is linear, or it is showing the same recovery, or the same concentration for all the dilutions. So ideally, the concentration or recovery of the sample should be similar for all dilutions. so as you have already noticed that most of the suppliers provide recovery and linearity data on the product data sheets. So besides that, the other important parameters to look for is the sensitivity and dynamic range, which is also provided on the data sheet provided with the ELISA kit. So the ELISA kits from different suppliers may have different parameters data. So I think it is always recommended for the end users to carefully compare these parameter data in terms of recovery, linearity data, or sensitivity, dynamic range to choose for the right ELISA kit. Next parameter is the detection system or the readout. So there are several different detection systems in ELISA kits are available. So that can be colorimetric, fluorescent, or luminescent methods. However, the principle is that all ELISA involves the immobilization of the analyte to the surface, as well as the use of enzyme and the matching substrate. So uh, the the analyte is involved is immobilized on the surface of the well, and then there is a use of an enzyme labeled antibody and a substrate. So based on that, one can use the detection system. So so it can be either colorimetric, fluorescent, or luminescent. So the most commonly used detection system is colorimetric. So the plate reader which can be used, but there are ELISA kits that are also available that can with the different detection system like fluorescent or luminescent methods. And last, the another crucial factor in determining or in selecting an ELISA kit is to look for the protocol, the experimental protocol. So ELISA kit with simple protocols, convenient operation, and short incubation time, it will definitely make the make it easier for end user to do an ELISA assay and to conveniently perform the assay to quantify their target protein into the samples. So just to give an example in this category can be a quick or easy detect ELISA kits that are available in the market nowadays. So in this like quick detect or easy these easy detect ELISA kits, the entire assay is relatively short. It can be completed in as little as 90 minutes. So these kits have less incubation steps, which can reduce, of course, the possible errors, and it has less washing steps, and that definitely helps to reduce the variations in the results. And that makes the entire protocol can be completed within short time, and it's very convenient, less less steps, because less steps are involved, so there are less possibility of manual errors or or variations in the results. So now once we know that, okay, so these are the tips that one should look for choosing an ELISA kits. So now based on these parameters, we want to know that how we can evaluate a kit. Like if we have an ELISA kit and we know, okay, now these are the parameters that we have to look. So the another step is to look like, okay, to look for how we can evaluate that this is an ideal ELISA kit for one's application. So I'm going to talk about like how we can, evaluate these parameters now. So the first thing to look for is the standard curve. So standard curve is very crucial if we want to quantify the concentration of target protein in the sample. So that means if we want to use, first of all, if we are looking for a quantitative ELISA, not quantitative ELISA, because we want to quantify the concentration of our target protein or analyte in a sample, so we should have to look for a standard curve. So this will also give us the, we should look for the linear range, and this will help us to calculate the actual testing or the concentration of our sample. Um, Along with this, standard curve also provide a dynamic range of the assay. Like in this example, you can see that this ELISA kit can detect target protein, and which is in these the range which the analyte can be detected is the higher range is 10 nanogram per ml and the lower is 0.16 nanogram per ml. So this is the dynamic range of the assay. So one should look for that the the expected concentration of their target protein in the sample should lie somewhere within this range. Uh, Next is the linearity of dilution. So that means the extent in which the spike dose response is linear. So as I have mentioned earlier, that linearity is it is the text index for experimental accuracy and specificity. So if we want to access the linearity of the assay, so how we can do that? The samples are usually spiked with high concentration of target protein using different matrices. So, and then these samples with different matrices are diluted with the sample diluent to produce samples with different values within the dynamic range of the standard curve, that even if there are different dilutions, but the the sample diluent have the concentration within the range of the standard curve. Um, so generally, these results are expressed in the percentage. So as you can see here that the samples are spiked and then diluted, and then you can see that the average recovery is almost linear or it's within the range. So that means the higher the recovery, the better the performance of the ELISA kit. Okay. So next in this, the next parameter to evaluate to evaluate an ELISA kit is to look for the precision. So precision determines the reproducibility of the experiment or of the assay. So it is oftenly measured in terms of CV, which stands for coefficient of variation. So the higher the precision, the better the repeatability. So generally, two types of precisions should be considered for ELISA assay, intra-assay precision and inter-assay precision. So intra-assay precision is the reproducibility between wells within the assay. So that means 2 wells or 3 wells or wells within the assay. Inter-assay precision is the precision, is the reproducibility between different assays. So this one should also look for the coefficient of variation or the or the reproducibility of the assays. So this information is always also generally provided into the data sheet. Next is the dynamic range and sensitivity. So as I already mentioned, the dynamic range is the upper and lower concentration of the target protein that the assay can quantify. So the lower the detection limit, the higher the sensitivity of the kit. So the better the sensitivity, the lower the sensitivity, the better the kit, and it is definitely preferable by the users. Next comes the specificity. So specificity is crucial index for ELISA kit as it confirms the cross reactivity and interference. So usually you have seen that suppliers, they provide all this information on their data sheet. So while manufacturing these ELISA kits, so it is always recommended that suppliers should select the raw material like antibodies with high specificity, just to ensure that ELISA kits has an excellent specificity. That means antibody should show a low, background or cross reactivity. And they should also analyze the cross reactivity and interference experiments to confirms that the target protein can only be recognized and detected, and it's not recognized any related protein. As you can, so this is an example that's how and where you can find all this information in the data sheet. So this is one of the example that I'm giving from one of the BioVision ELISA kits data sheet, that see how we mentioned here: detection range, sensitivity, specificity, and if there is a cross reactivity, and this is a precision or coefficient of variation. So this is one typical example. Then last is the recovery. As I already mentioned, it determines the experimental accuracy and specificity of the experiment. So it also reflects the loss of the target protein in the sample analysis process. So as I mentioned earlier, the higher the recovery, the lower the higher the recovery, the higher the accuracy. So as you can see, the higher the recovery rate, the higher the performance or the accuracy of the ELISA kit. So with this, I think I covered all the the crucial tips how end users can select an ideal or perfect ELISA kit or appropriate ELISA kit for their application. When they found the kit, how they can evaluate the kit, whether it is it is the kit that they want to use for their application. The kit is reproducible, it has high sensitivity, or it has, the kit is accurate, it is specific to the sample. So these are the parameters that one should look for while selecting an ELISA kit for their experiments. So, moving forward with that, I'm going to talk about some of the key features of our BioVision ELISA kits. So all BioVision ELISA kits are, they are QC'd for look for different parameters. So I'm going to talk about that, some of the key features and high selling points. So all of our ELISA kits are highly versatile. They are available in different formats like sandwich ELISA or competitive ELISA. We offer ELISA kits for different sample species like human, mouse, rat, as well as we also offer ELISA kits that can be used for any species, like universal, that can be used for any sample species. Our ELISA kits are, tested for accuracy. They show reproducible results with low intra and interassay variability. They have high sensitivity and specificity, and provide very low background. That means, there is no cross reactivity. And we make sure that our ELISA kits showed minimum lot to lot consistency. Uh, uh, our ELISA kits are validated on biological samples. They provide best results on broad range of sample types, like serum, plasma, different biological samples, if possible, food samples. It depends upon the targets. If the ELISA kit is validated on biological samples, it definitely ensure that it reacts with endogenous protein. So this is also one of the parameter which end user can look for, that if the ELISA kit is validated on biological sample or not. Our ELISA kits are very convenient. So the protocol is very simple with short experiment time. They are available or in 96 well format, ready to use, and mostly they comes in a micro well strips. That means which allows the end users to use as many wells as they want in a particular experiment, and they can save the other wells or the other strips later for for future use. So that reduce any wastage, and it makes the ELISA kit very cost effective. And most of our ELISA kit, or almost all of our ELISA kits, are available in colorimetric format. So the readout is on it can be read on 400 nanometer wavelength. So only a simple it requires only a simple plate reader. No expensive instrument is required. Uh, and one of the most important, or selling feature of our ELISA kits are they are very affordable. Our pricing are very competitive in compared to the similar kits available in the market from other suppliers. And also we offer a wide selection of targets. We offer almost 500, more than 500 ELISA kits available for various research areas. So I'm going to give you a brief overview about, these are the research areas on which we offer ELISA kits for. So these they are we have categorized into very small broad categories, just to give an overview. If you want to, you know, if you want to know that our ELISA kits for which research areas we offer ELISA kits for. So this is a broad overview. So we offer ELISA kits for cancer research, which includes ELISA kits for the research of angiogenesis, immune checkpoint, tumor biomarkers. Then we offer ELISA kits for immunology research, which includes ELISA kits for the detection of cytokines, interleukins, immuno-oncology targets. We also offer ELISA kits for the detection of immunoglobulins isotype, like for example, IgG, IgM for different species. Um, and we have a very broad range of ELISA kits for toxicology research, which includes ELISA kits for antibiotic research, for the detection of mycotoxins, for the detection of environmental toxins, food toxins or food additives. And I'm going to talk about this category in detail in few minutes. Uh, uh, we also offer ELISA kit for the detection of various infectious diseases. This include ELISA kit for the detection of different viral diseases, bacterial diseases, and also I'm going to talk about this one in details. Next, ELISA kit category that we offer is the metabolism, which includes ELISA kit for the detection of different targets that can be used in the research of diabetes, uh, drug metabolism, hyperthyroidism, obesity, and these are just a name, just to name a few. Metabolism is a very, very, very huge category, and we have many, many, many ELISA kits in this category for many more diseases, and you will get the more information if you can check on our website. But we have this is a very huge category for which we can offer ELISA kit for. Next is we also offer ELISA kit for stem cell research, which includes ELISA kits for the detection of different biomarkers. I'm going to talk about this one also. ELISA kits for the detection of different differentiation markers and proliferation. We also offer ELISA kits for neuroscience research, which includes the ELISA kit for Alzheimer diseases, for the research on Alzheimer diseases, neurotransmitters, and Parkinson diseases. And last but not the least, very hot topic, we do offer for ELISA kits for immunotherapy research, which includes a very, very hot topic, biosimilar research, ELISA kit for the detection of biosimilars. And I would like to mention here that this is one of our high selling category of ELISA kits, biosimilars. Yeah, if you want to make a note, just make a note. This is our best selling category of ELISA kits. And also we include in this one ELISA kits for the detection of immune checkpoint. Okay, so moving forward, I'm going to talk about few different categories here, which includes toxicology, infectious diseases, and biomarkers. So let's start with the toxicology. So toxicology, again, as I mentioned, that we have categorized into different topics. We have ELISA kits for the detection of antibiotics, mycotoxins, environmental toxins, or food additives. So let's start with the ELISA kit for the detection of antibiotics. So antibiotics, as we all know, are also known as antibacterials. These are kind of drugs that destroy or slow down the growth of bacteria. So generally, these antibiotics are can be given to animals for the treatment and prevention of diseases and growth promotion. So these ELISA kits can be used for the detection of antibiotic residues either in food sample or in human samples. Um, so some of the good ELISA kits are mentioned here, the list in this list, and as you can see that they all have a very high sensitivity, so they can detect these antibiotics in a very small concentration, like as in nanograms. You can see in almost in or zero point 0.1 nanogram, zero point 0.3 nanograms. And they can be used for a wide range of samples. They can be used on human samples like tissue, serum. They can be or different biological samples, urine. They can be used for different food samples like milk, egg, milk powder, honey. So different types of samples, wide selection of samples. So uh, and of course, as I mentioned earlier, some of these ELISA kits are validated on samples. You can see here, like the spike experiment. So this is the data from one of our gentamicin ELISA kit. That this is a ELISA kit that can detect gentamicin in serum or urine, and in other samples too. So you can see that it is the gentamicin concentration is detected in milk samples, serum, and urine. Actually, this is a data that's showing the spiking experiment and the recovery. So you can see that this can be used for different types of samples. However, I would like to mention here that this is our gentamicin ELISA kit, but we have a different gentamicin ELISA kit that can also be used for, food samples as well as serum and serum and plasma samples, and this is one of the high selling product in this category, gentamicin ELISA kit. Please try to remember this K4206. So if you have a customer who would like to have ELISA kit to detect gentamicin in their samples, I would highly recommend this kit. And because we sell thousands of this kit, so far, and we have never received any complaint, this is our, the top selling product, gentamicin K4206. Besides that, some other ELISA kits, some other popular ELISA kits are kanamycin ELISA kits, ampicillin ELISA kits, streptomycin ELISA kits. These are also our good selling products. So moving forward in this category of toxicology ELISA kits, the another ELISA kit is the ELISA kits for the detection of mycotoxins. So mycotoxins are low molecular mass secondary metabolites that are produced by fungi. So these molds, they can produce or they can grow on different type of foodstuffs like cereals, dry fruits, nuts, and species. These metabolites, when we ingest or inhaled or it get absorbed through skin, they can cause different kind of diseases or death in in humans or in animals. So some of the agriculturally important mycotoxins are aflatoxins here, or fuminosins, or this OTA, ochratoxin. So these are some of the very agriculturally relevant toxins that are real an issue in the field of agriculture. Um, so and this is the list of the top selling products in this category. Again, they can be used for a wide range of samples, food samples. They all have, as you can see, they are all very sensitive. They can have a very low sensitivity range. You can see they can detect as as low as 1.1 nanogram or 0.02 nanogram of these toxins in the samples. So if you, this is a very niche product, and if you would like to know the customer base, so the customer who would be interested in these kind of products would be researchers who are in the agriculture industry, or who are doing research in plants. Especially they would be interested in this in this kind of in these ELISA kits or in this product line. Uh, so next in this toxicology ELISA kit category is the ELISA kits for the detection of food additives in the sample. So as we all know that the food contamination with biological and environmental pollutants can occur through several stages of food production and processing. Food additives have serious effects on health. These ELISA kits can easily, they can easily detect the chemical toxicity or the toxicity of these chemicals in the food samples. Some of the good selling kits in this category are this salbutamol or these sulfonamide residues. Almost all the kits listed here are good selling kits, and they also have a very high sensitivity, and they can be used for a broad range of sample types. And next in this category is the host cell proteins, toxicity of host cell proteins. So as we all know that host cell protein, HCPs, are widely used in the expression of therapeutic proteins, for example, CHO cells or E. coli cells. So however, after the expression, HCPs can still be present at significant concentration after following several purification steps, and can even be co-purified and concentrated with the drug, its substance itself. So which is definitely somebody can be toxic for the for various downstream applications or therapeutic applications. So these ELISA kits are very helpful in detecting the host cell protein in these biological samples, especially cell lysates and other different biological samples. So you can see that these samples can be used for cell lysates or other biological samples like serum, plasma, urine, and they have a broad detection range, 0.1 to 100 nanogram, or 1.5 nanogram to 60 nanograms. So high detection range and low sensitivity. So along with these host cell proteins, CHO host cell proteins and E. coli protein, we also offer ELISA kit for detection of BSA in the sample. Although this is not a host cell protein, but this is also one of the, I wouldn't say toxic substance, but a contaminant that always present in the sample, and researchers they always don't want to have it because it can definitely affect the downstream application. So this is also one of our good selling product, BSA ELISA kit that can be that can detect the BSA in the sample, in the human biological samples. Okay, so the last category in this toxicology ELISA kit category is ELISA kits for you can say environmental toxins like bisphenol A or melamine A. Bisphenol A, we all know that it's a BPA. It is a very it is a huge problem nowadays for our environment because it's present in plastic, and through these plastics waste, it's getting into our food, it's getting into our body, it is affecting the health and causing serious issues, health issues. So we do offer a kit to detect BPA in different biological samples like serum and urine, and you can see here that's the data that we have shown is it's tested on samples, serum samples and urine sample, spiked sample. So this is also one of our a good selling kit. And melamine is also present in plastic, and it can be also used for different human samples as well as different food samples. So these all, they also have a like high detection range or low sensitivity. So with this, I would like to conclude, our toxicology, ELISA kit for toxicology, or this category of ELISA kits. And next, I would like to move to the biomarker. So biomarkers, we know that they are generally referred to the measurable indicators of some biological state or condition. So these biomarkers are the perfect choice for early detection, screening, and diagnosis of any disease. Uh, we offer a wide selection of ELISA kits, which is a very simple and cost-effective method for detecting or quantifying these biomarkers in different samples for research. The different application for this ELISA kits include that they can be used to understand the mechanism of action, safety or toxicity indicators, in different disease conditions. They can be used as a diagnosis tool. They can be used to for the selection of lead candidate. These ELISA kits can be useful in the screening of tumor biomarkers or in measuring the hormone levels. So some of the key features are uh, all these kits are very accurate with reproducible results and with low intra and inter-assay variability, high sensitivity, high specificity, and low background, and they can be used for broad range of samples. Uh, so as you can see here, this table shows the list of ELISA kits that are available to detect targets in different disease types. So, like for example, this anascreen and 3NT these are the biomarker for the autoimmune diseases. So these are the different, the catalog numbers of the ELISA kits that are available for the detection of these biomarkers. We do offer CRP protein detection kit, which is a biomarker for inflammation, NSC, which is a biomarker for neuronal damage, creatinins, cytosine C, NGL, it is a biomarker for renal diseases. So this is the list of the diseases and the list of the biomarker that we do offer, for the detection of these diseases. So this is the list of some of our good selling products. And next is our category of ELISA kit category of infectious diseases. So we all know that infectious diseases, which are also known as transmissible diseases, are disorders caused by various pathogens such as bacteria, viruses, fungi, and parasites. These infectious diseases, they cause around 15% of total death in the human population. So these ELISA kits can be used to detect and to study the mechanism and prediction of these diseases, these infectious diseases. That those are the applications usually for the detection or to study the mechanism or the prediction of these diseases. So, some of the key features of these ELISA kits are, they have high sensitivity. They can detect IgG, IgM, and other immunoglobulins in the samples. And they are very useful in detecting antibodies, induced by these infectious diseases in response to the host immune system. Yeah, so this is the list of ELISA kits available for detection of various immunoglobulins in different diseases. And one recent addition in this category is SARS-CoV-2, as we all are very familiar and very much interested nowadays in knowing, about this coronavirus. So we have recently launched this kit that can detect coronavirus antibodies in the biological sample. So this is a qualitative ELISA kit that can detect the presence of these antibodies in the human sample. And it is very helpful to understand the disease progression, and it is helpful for the researchers to find the therapeutic again to find the therapeutics against this disease. Along with this, although I have not mentioned here, and which is not related to this particular category, but with the coronavirus, is also we do offer ELISA kit for ACE2, which is a key player and involved in the infection process of coronavirus. So which is also now, you know, many of our customers are interested in this kit also, as it is involved in the infection of coronavirus. So so just to mention that, this kit is getting very popular nowadays. And I also would like to comment that our R&D team is working on other assays that are related to coronavirus, so they are working for working on ELISA kits and or several other inhibitor screening kits. So in case if you have customer you are interested, please feel free to reach out to us, or definitely when these kits will be launched, they are in our pipeline, in the last almost in the launch process. And when these kits will be launched, of course you will get the notification. But in case if you have any questions, or if you want to know that, if your customer they have any particular interest, or they want to know any they are interested in any particular kind of kit, maybe it's the one that we are currently developing, so please feel free to contact us. Yeah, but just stay tuned. You will be informed soon with these new additions. Okay, so I would like to conclude my presentation with some, tips. This is basically mainly for the, end users, or for those, or who all are involved in the tech support, and they frequently get these, complaints from the customers that, we are doing our essay, but the ELISA kit is not working for us. So I would like to go, just I would like to share some tips that they can definitely, share with their customers that can be helpful in their troubleshooting, or, just to know why their experiments are not getting the appropriate results. So the most common, so I would like to highlight mostly on two basic problems that are, usually we get these complaints, is either low signal, low sensitivity, or high background. So the possible reasons for getting no signal is could be that they are not incubating for enough time, or the maybe the end users, the interest of the target, the target protein is present in a very low concentration, so below the detection limit of the assay, and the assay is not capable of detecting it, or maybe they are diluting their samples too much, dilution is too high. Maybe they are using the wrong kit, which is not compatible. I mean, they have a different species and they are using a different kit which is not compatible for their species. They are not adding enough detection reagent or substrate in the sample, and hence the readout is not appropriate, or giving lower OD, or not giving a high signal. Maybe they are not incubating for the appropriate time, the incubation time is short. Maybe that the washing, after washing steps, the wells dried out, which we don't recommend because that can significantly reduce the signal. It can also be due to that they are not reading their plate on the correct wavelength, that's appropriate for the assay. Also, the low sensitivity could be due to the preservative in the samples. Maybe the samples, some like sodium azide, it can interfere in the samples, can interfere with the signal, with the HRP. So preservative like sodium azide is definitely gives them a low signal, and it's not recommended to have it in the sample while using these ELISA kits. Maybe they are using wrong filter, like incorrect wavelength, or wrong filter to read their plate. The possible reasons for getting high background is that the wells are insufficiently washed, and still there is antibody present in it, or substrate present in it, that is giving them a high high background. Maybe they are using too much detection antibody, the dilution is not appropriate. Or it could be they are getting high background because they have incubated the plate for too long with the substrate or in the light, or the incubation time is too long before adding the stop solution or eating the plate. It could be that they are getting high background because they have not stored their sample, their ELISA kits properly, and it is already exposed or dried out, or the plate is dirty or have some precipitate in it. So last is, we highly recommend to follow these steps to get the appropriate results and to get the high accuracy of the experiments, is to find the right ELISA kit, to find the right target, sample types, detection range. Always run pilot experiment and standard curve, like look for detection range, look for sensitivity, specificity, spike recovery data, linearity of dilution test. As you can see here, that this is one of the uh, protein range, like this is how it can be defined: high, medium, low, very high, and these are the different dilution factors and the recovery. So look for all these data on the data sheet. Use fresh samples and ELISA kit within the expiry period. Always run assay as soon as possible after sample collection. Always run standard curve with each experiment. Do not use one standard curve for every experiment. Use proper controls, avoid freeze thaw cycles of the samples if they are frozen, and run all samples in replicates. So with this I would like to conclude my webinar, and this is the available literature for ELISA kits. You can find all these flyers and brochures on our website, and if you don't, then just feel free to reach out to us. And you can reach out to us, either this is my email address, or you can always reach out to us on our tech support email address, which is tech@biovision.com, and I am open for any questions if you have. Thank you very much again for attending. Please let me know if you have any questions. Can you hear me? Hello? Can you please raise your hands if you can hear me? Thank you. No questions? Okay, so if we don't have any questions, we would like to conclude this session here. And you can always reach out to us on our tech support email address, or this is my personal email address. If you have any questions, we will get back to you. Okay, thank you, everybody. Thank you so much for attending. Okay, so thank you everyone for attending. I hope you have enjoyed this webinar, and if you have any questions, please. Okay, yeah, we have a question. If I can share the PPT, the presentation, definitely, yeah. So we will share the presentation, yeah. So on to all the attendees, definitely, yeah. We will share the presentation. So all those who have registered for this webinar, they will get the present copy of presentation.